Hello and welcome back to our discussion of sensory systems. Next up, we're going to be talking about olfaction and gustation, which are our fancy scientific words for smell and taste, respectively. Okay, so starting with smell. Smell occurs at kind of the top of our nasal cavity here. Okay, at the top of our nasal cavity, we have a structure called the olfactory epithelium. And that is the structure where the initial detection of odorant molecules, or the molecules that we're smelling, occurs. Okay, let's take a closer look at the olfactory epithelium. Just to orient you, again, this is the bone that's at the top of the nasal cavity. We have our olfactory epithelium right here. And then at the bottom here, we've got a mucus layer, okay? The olfactory epithelium consists of these blue cells, olfactory sensory neurons. Those are the ones that are going to be detecting our odorant molecules. And then we've got these support cells in pink. The olfactory sensory neurons extend dendrites into the mucus layer here. The way that we smell, the way that we detect odorant molecules, is that the odorant molecules come up here and they dissolve in the mucus layer, and then they are detected, they bind to receptors on these olfactory cilia, the dendrites of these olfactory sensory neurons, okay? Sorry, there you go. That triggers, like when we have the binding of the odorant molecule to receptors on these olfactory cilia, that leads to a G protein mediated cascade that leads to ion channel opening. And when we have ion channels open, as you know, we're going to change the membrane potential, and that is our electrical signal. Okay, so that electrical signal is going to be, well, eventually it's going to be an action potential that goes down the axon of the olfactory sensory neuron, and it's going to be carried to the brain for further processing. Okay, each olfactory neuron only expresses one receptor, which means that it can, for the most part, detect just one molecule, or maybe some other molecules that are similar to that molecule. So just a handful of odorant molecules. Okay, let's move on to gustation, or in other words, our sense of taste. As you are probably aware, taste occurs mostly through our tongues, okay? Our tongues have these structures in them called taste buds. Also probably not surprising to you. But what you can see in this picture on the right is the structure of a taste bud. Okay, so this is if you like took your tongue and cut it like this. Okay, so a taste bud consists of a sort of a collection of cells, okay, that are kind of bundled together. And they're exposed to the outside. So they're kind of like buried here in the tongue. Okay, so they're embedded here, and they're exposed to the outside, the rest of your mouth, through this structure called the taste pore. Okay, these cells are not neurons, they're epithelial cells. They're like modified epithelial cells. Okay. So let's take a closer look at those epithelial cells in cartoon form. Okay, so here's a cartoon form of a taste bud. So a taste bud usually has about 50 to 100 of these epithelial cells in them, our taste receptor cells. But, you know, for simplicity, only a few are shown in this picture. Okay, so what do I want to say? So there are these epithelial cells here, they're bundled together. A couple features that I want to draw your attention to up here are, one, we have these microvilli at the tips of each of these cells to increase contact with the saliva and any tastent molecules that we might have there. We've also got these tight junctions here between the cells, okay? So the way that we taste, at least for these three taste sensations, sweet, umami, and bitter, which I'll talk about in a little bit, is that these are molecules that bind to receptors on these cells. That leads to a G protein coupled, a G protein mediated cascade. And then eventually that leads to a graded potential that then releases neurotransmitter onto these neurons here, okay? What else do I want to say? Here's a question for you. So why is it useful to have tight junctions here? Why do you think it would be useful to have tight junctions here? Just kind of think about that for a second. Okay, so I don't know what you came up with, if you had any ideas, but one of the advantages of having tight junctions here, remember tight junctions are going to block off this space between the cells, okay? So if we're blocking off that space, that keeps molecules up here, which is like the top surface of the tongue up here. And that's useful because that's where the receptors are for detecting those molecules. So the tight junctions keep those molecules up here so that they can be detected, okay? Couple more things I want to say about the sensation of taste is we have 5 official taste sensations: sweet, sour, bitter, salty, and umami. So umami is kind of savoriness. It's like the taste of amino acids. We don't know, like as a scientific community, we don't know that much about taste. We're constantly sort of identifying new potential taste receptors. So we think right now that there might be taste receptors for carbon dioxide, for certain fats, but not enough evidence yet to really come to a conclusion there. The other thing that I want to say is that most of what we taste or what we think we're tasting is actually our sense of smell. So you probably know this from when you've had a cold and you haven't been able to smell anything. When you're eating, you barely notice the taste of what you're eating, right? Your taste sensations are really just these 5 sensations: sweet, bitter, sour, salty, umami. All those other experiences that we have while we're eating come to us through our noses. So those are all volatile molecules, molecules that are floating around in the air that come up through our nose and are detected by our olfactory epithelium.